great looking crowd. As Melissa said, my name is Rafael Rosengarten. I'm co-founder and CEO of Genialis. Before this, in fact, immediately before this, I was a Keck Fellow and at Baylor College of Medicine. Today, I want to really leave this open for dialogue. So if you do have questions, put them in the chat, and maybe Melissa, either you or someone can keep an eye on whether they're like an urgent question and I should shut up and address it, or whether we can hang on to it for the end. I'm also going to try to leave some time to just have a dialogue at the end. What I love about the Keck Seminar and the Keck training program in general is it's, I think, meant to help open people's eyes to kind of the world of possibility about careers in biomedical informatics and data science and the intersection of biology and data. And that's something that I'm really passionate about. And so I'd love to just be able to have a little bit of conversation at the end. But without further ado, I do have some things planned to talk about today. So hopefully we can do that. Just bring up my slides here on my other device. All right, so just a few topics. I'm going to start by giving a brief history of my own kind of trajectory into this. And I call myself an accidental entrepreneur, and I really think that's a fair designation. I'm going to spend probably the bulk of my time on any one subject talking about Genialis and what we do. Oh, I'm getting ahead of myself here. That's probably the thing I know the best. Obviously, it's my company, and it's something I spend a lot of brainwaves on every day. And what we do is computational precision medicine. And then I have some thoughts on artificial intelligence in healthcare, which I know is not exactly the same thing as what GCC Keck is focused on. But you know, if you're going to pick a buzzy buzzword to sum it all up, you might as well call it artificial intelligence in healthcare. So we'll spend some time there. Why do I call myself an accidental entrepreneur, though? So the reason why is because I didn't know what I was doing when I was getting into it. I'm still not entirely sure I know what I'm doing, but I certainly didn't know what I was doing when I was getting into it. And I was a diehard academic, or at least I thought I was until I wasn't anymore. That's me on the left. I have the same crappy haircut and 5 o'clock shadow, pipetting some stuff in Gaddy's lab. I think I was posing for that picture. I would never hold a plate up like that to pipette. And so I was at Baylor from 2011 to 2015 and had this really, really wonderful opportunity while I was at Baylor to collaborate with a research group from Slovenia. And the reason this was possible is Dr. Blaž Zupan, who runs a data science group in Slovenia, also happens to have an adjunct or associate position at Baylor. I don't want to steal his title, but he's got something at Baylor. So he comes over every year. And if you are at Baylor and happen to have the opportunity to take one of his courses, I think they're usually in September, definitely do that. Blaž is great. He's brilliant. He's a dedicated teacher, one of my favorite mentors. But he would come over to Baylor and he would bring graduate students to kind of, bring their data science to the lab and see what the lab folks needed to do with data and get the creative juices flowing. And so I had the distinct pleasure of working with one of his most brilliant students, a woman named Marinka Zitnik. She then went to Stanford and is now a professor at Harvard. But with Marinka, we were interested in figuring out how we could integrate lots of different data sets together, different data of different types and flavors, and then make predictions based on what I call the totality of evidence. And I actually gave a plenary session talk at the NLM meeting in Pittsburgh, some number of years ago, on the work that it was really me taking credit for Marinka's work, but on the work I was doing to collaborate with Marinka. And so I drank the Kool-Aid. I was all in. The predictions from Marinka's algorithms were shaving like years off of functional genetic screens that we would have to do in the lab. Like, why do the functional genetic screen if you could just predict what genes were relevant, and then you could make those mutants, and sure enough, like 80% of them were actually spot on the phenotypes you predict. So I thought this was the coolest thing ever, and I wanted to do something with it. Right at the end of my Keck fellowship, another one of Blaž's students came over, and this was a guy named Micha Stajdahar. And Micha had just finished his PhD in artificial intelligence, something to do with networks. And he had also just started a company. He'd started Genialis with a couple other Slovenian dudes. The other guys were not data scientists, and they weren't biologists either. They were just wannabe entrepreneurs. And I mean wannabe in sort of the most derogatory sense possible. And so they had started this company and didn't have a clue what they were going to do. They were going to commercialize some software out of Blage's lab. They were going to do something in the biology space, but none of them could find biology with two hands if they looked for it. And so Micha came to Baylor on what I would call an industrial postdoc to figure out what their technology could be used for. And so we worked really closely together. And 1 year into his 2-year postdoc, I committed and joined Genialis as an early employee. I joined by joining the executive group. So I was chief product officer, head of software product. I negotiated near founder level equity, and I spun up a Delaware C corporation, which I'm happy to explain what that is later. And the Delaware C corporation then took ownership of the existing Slovenian company so that we would have sort of the right structure for being a company in the world. And so that's how I became an entrepreneur. And literally, I got off the phone with my new boss, my CEO, and he said, "Yeah, I was thinking you'd be the chief product officer." And I sat on my couch and Googled, what is a chief product officer? Like, I truly didn't know what I was getting into. But what I did know is that we wanted to use data science to solve hard problems in biomedicine. The other thing I knew is that to do that, you needed the data, and getting all the data you'd want into a usable format was really, really hard. It was, damn near impossible. And so that kind of set our course. We figured we're going to build software to corral the data, to harness the data, and then we're going to apply it to some interesting problems. But until I kind of jumped into this, and my wife's only criteria was, yeah, you can do this, but can you just not make less than you did as a postdoc? That was her only request. So try not to earn less in your first year as a 30-something-year-old of having a real job. Just try not to earn less than you did as a postdoc. So that was my goal. But seriously, until then, I was an academic, except I wasn't a very good one. Published some, but not terribly high profile. But even like those little images at the top of the screen, those kind of cool pictures of cells and organisms, those are plankton. Those are images that my company has since licensed from my cell biologist uncle, who is my personal scientific mentor, and whose book on plankton I edited, because at the end of the day, I was a natural historian. I just like biology. And here I am now, anyway, the CEO of a biotech. We've got 30 employees. We make, not lots, but millions of dollars a year, enough to support 30 employees. And we build important computational products. So it really was, in my recollection, by accident. But let me tell you a little bit more about what Genialis does. So computational precision medicine is how I kind of define the field. What I mean by computational is that we're a dry lab company. So we don't have wet labs currently anyway. We don't do our own genomic sequencing. We don't have cell lines or mice. We muck around with the data. This may change eventually, but for today's purposes, that's what we are. And precision medicine, as I'll define it a little bit, is sort of this idea that you can treat people or intervene in people's disease based on some very specific knowledge of the nature of their disease, right? So you're really matching the therapeutic intervention with the disease. And what I'm going to do for this section is actually walk you through some slides that I put together for a talk 2 months ago, almost 3 months ago. At the beginning of November, we launched this product, Genialis Responder ID. It's our second branded product. The first one was that software to corral all the data. That was called Genialis Expressions. Genialis Responder ID is a machine learning enabled biomarker platform. It'll also become a drug discovery platform in the future. And so I gave this talk at Biodata Basel, which used to be pre-COVID was sort of one of the industry bellwether events where all the tech companies would come and show off your shiny new thing and hang out in Basel and spend way too much on crappy food and drink. It's one of the most expensive cities in the world. Anyway, biomarkers are absolutely essential for precision medicine. Absolutely essential for precision medicine. Biomarkers are how you determine the nature of a patient's disease. Back in November, these numbers are a little old. I did a PubMed search. This was the morning of the talk, and there were over 1 million PubMed entries for the word biomarker. That doesn't mean there were 1 million biomarkers, but orders of magnitude. And that had been increasing exponentially for the last 2 decades. And then if you go over to the FDA's website and you search for companion diagnostics, there were 48. There are 48 approved companion diagnostic devices, 48. But there are 1 million papers purporting to have found a biomarker. Nucleic acid tests is a little bit better than companion diagnostics, but the point is there are orders and orders of magnitude fewer actual tests we can use on actual people to figure out what the hell is going on than there are efforts to find those, right? There's a really, really narrow funnel there. And so this is important because, again, biomarkers are key to precision medicine. We use them for all sorts of things. And now, just to clear up some nomenclature, a biomarker is a pretty generic term, just means something you can measure that reads out the biology. We use them for things like telling whether tissue is disease or normal. This is actually pretty exciting. I stole the little picture on the left, the pink pathology slide picture from a company called Page.ai, and they just had the first pathology artificial intelligence tool, I'm not sure what I would call a product, cleared by the FDA for determining tissue normal. So that's pretty cool. We use it for prognosis. So for those of you who work in the cancer space, especially maybe at MD Anderson or elsewhere, you probably know a lot about sort of, risk-based scores, determining, which groups of patients are more likely to do better or worse, how serious is your disease. Prognosis is important because you can make treatment decisions like how aggressively to intervene or just to surveil. Biomarkers are used in pharmacology. So, what is the drug doing in a system, in people or whatever your system, monitoring disease after therapy, et cetera. And then prediction. And prediction is really where I'm most interested, is what Genialis spends the most time on. Prediction is saying, can we actually figure out or predict ahead of time? Can we build a model that will tell us which individuals are most likely to respond to a particular therapeutic intervention and which ones are not? And this is important for a number of reasons. If you can predict the winners, this is meaningful in the real world in clinical trial settings. For example, in a gastric cancer cohort, this was a retrospective analysis my team did, we were able to show that if you had enriched using the biomarker we built ahead of treatment, the effective response rate would have gone up from 18% in the unselected group to 44% of patients that you selected. And that enrichment is better than a lot of the biomarkers that are currently used for immune checkpoint inhibitors. It's the class of drugs we were looking at, and that's about as good as you get in this disease indication. And so what that would mean for those patients is many fewer patients would get a drug that's not going to help them, that has severe adverse effects, and that costs a shitload of money, right? So it would be much more efficient to do it this way. And if you happen to be a biotech or a small pharma company developing a drug, that drug is 5 to 12 times more likely to progress from one clinical trial phase to the next if you're using a biomarker in the trial. And that doesn't even, that's like irrespective of the kind of biomarker. It turns out in the study that showed that wasn't our work, that was another group that published recently, the investigational biomarkers did even better than the approved ones. And what that suggests to me is that the ones that are trying to tackle complexity that haven't been approved yet are working better. The reason this is so hard, the reason why there are 1 million PubMed entries and 48 FDA approvals is because biology is hard. And I know that sounds kind of glib or trite, but it is. I'm showing you this ridiculous subway map on the right, and that's an old 2005, 15-year-old pathway map for the EGFR pathway. So we probably know it's even worse than this. So imagine you're trying to drug EGFR, okay? And you want to guess which patients are going to respond to that drug. How on earth are you going to abstract this subway map into something that you can measure robustly, repeatably, reliably in patients at the point of care? Well, the FDA hasn't figured that out yet. Of those 48 approved companion diagnostics, 41 of them only measure a single molecule, right? That's not going to work. And in the case of something like EGFR, they're probably just looking at known mutations in EGFR, which may be necessary, but are not sufficient to predict response. We know that's true of almost all targeted therapies against driver mutations. 7 of those FDA-approved devices measure multiple analytes or multiple variables. But those aren't what I would call complex signatures. Maybe the closest would be like tumor mutational burden, but that's still just kind of a proxy for neoantidote load. There are no approved devices that measure complex signatures or multimodal markers, so where you're measuring different ohms or different layers of the onion. And I think that's going to change. In fact, I'm quite certain it's going to change, and I hope that my team is among the group that helps change it. But a big part of fomenting that change is figuring out how to abstract the complexity. And so we need to embrace it. We need to run head on into that fire, right? So there are three pieces that I think we need. And these are well underway. This isn't fanciful. This is just sort of stating the obvious. You need high throughput data generation, right? So you need enough data to model. You need enough data to get signal from the noise. And these data can come in a lot of different flavors. So I was recently interviewed on a podcast, and the podcast guest asked me to kind of come up with a working definition of next generation sequencing. And I told him I thought it should be called this generation sequencing because we've been doing it for a long time now, thinking about things like Illumina short read sequencing, whether it's whole exome or RNA or whatever. But it's not just omics. I like omics. That's mostly what my company works on. But high throughput image generation, image analysis is really in vogue right now. I have a photo credit here to Recursion Pharma. If you don't know Recursion, you should look them up. A super cool pharma company out of Salt Lake City, where Melissa and I spent a really lovely Keck retreat once. And Recursion has even open sourced one of their early data sets. This is like terabytes of image data. And I think they built a Kaggle competition around it, or some sort of open source data mining competition. Super cool stuff with image analysis. But anyway, we need high throughput data generation, and I'm one of those kind of all of the above kinds of guys. You need software to deal with it. And so this is where Genialis got its start. I'm partial to our software for omics. It does a good job. We've partnered out with groups like Roche and Thermo Fisher in the past, so it's like legitimately good software. But you can use others. You could build your own. I don't recommend it. And obviously, depending on the kind of data, you would use different kinds of software. And the last piece is we need this, what I'm calling here, new mathematics. I'll be honest, I don't really care if it's machine learning, although I kind of do, because I think machine learning, which is already a pretty broad class of algorithms as a subset of artificial intelligence, the way that I think about it as a non-data scientist, as a wet lab guy, machine learning are algorithms that are essentially conceived of and designed to recognize patterns, right? And when I talk about abstracting complexity, that's what I mean. I mean finding patterns that we humans would not find with our own two eyeballs or our own intuition. And so what I want to do next, before I get ahead of myself, is talk a little bit in concrete terms about three challenges that I believe are partly to blame for this giant gulf in coming up with, we'll call them signatures. And again, a signature could be a gene expression signature, but it could be a lot of other things. But some sort of signature of disease in a research setting versus getting that to the point of utility in a patient. And so I'm going to show you three pairs of slides. Each one is a description of the challenge and then a description of one possible solution that we've been working on. And this is just to kind of drive it home and make it concrete and hopefully not too many eyeballs glaze over. So the first one is thinking about our goal of predictive biomarkers. We want to model drug response, right? And so the way a lot of groups go about this is by trying to model drug response directly on the responders. Imagine you have a clinical trial data set and some of the patients did well. You want to figure out what do those guys have in common, right? What are those patients? What is the signal that made those folks stand out from the others? I think this is really impractical. I think it's inadvisable, and I'll tell you why. Those data sets tend to be too small. They're not always too small, but they tend to be too small to do good modeling. Observational trials have a median of 200-ish, 229 patients. There are at least 20,000 expressed genes in the genome. There might be 50,000. It depends on how you like to count. And so if you just tried to build an unsupervised model and you put 20,000 genes in, well, you're overfitting by orders of magnitude. You want to have more patients, fewer genes, not the other way around, right? So the data sets just tend to be too small for that kind of approach, in my opinion. The other challenge is the categories that you're trying to model are lopsided. So I'm showing you a couple different data sets that we've worked on. One is from a phase 1 trial in mesothelioma, another's a phase 2 trial in melanoma. different diseases. And one thing to appreciate from the figure on the left is, again, there just aren't that many patients. I think there are 30 something patients there. And the way this graph works is the samples are arrayed left to right on the x-axis. The y-axis was some response probability, a predictor that we tried to build, which did not work. And the colored sort of groups are the response. So PR, the green group, those are the ones you want. Those are the partial responses. Everyone else either stayed the same or got worse. And so there weren't enough patients to do this. And furthermore, there were only about a quarter of the data set were partial responders. And so this is imbalanced. You're asking an algorithm to learn something that the null model, the random model, is going to do better at. And the same story is generally illustrated in a different kind of plot on the right. Here we have 4 response categories. We have complete responders as well. But as you can see, virtually every patient is all the way on the right in the progressive disease. So this is just kind of a fool's errand. The approach that we take at Genialis, rather, is to model the underlying disease biology, which is hard because you have to have biologists on your team. You have to think really deeply about the disease domain and have a therapeutic thesis. So I'll try to illustrate this again with a bit of a real world example. On the left is a figure from a seminal review article by Dan Chen and Ira Mellman. Ira was my cell biology professor once. He's at Genentech now. And they were trying to simplify, come up with, again, an abstraction of what's going on in the tumor microenvironment that either predicts success with a checkpoint inhibitor or predicts failure. What I want you to take away from that figure on the left is just it's complicated. That's all I'm asking you to see there. There's a lot of colors, there are a lot of boxes, there are a lot of concentric rings. They tried to make it simple, and what I'm telling you is there's a lot of shit going on. And so we took a different approach with a company called Ancserna Therapeutics. These are some close collaborators of ours in the industry, where we rethought about sort of the biology depicted on the left as 2 axes. We said, all right, we know immune biology is important, and we know that angiogenesis in the tumor microenvironment, the stroma, is important, right? So you've got the immune system, which you want to get activated and fight cancer. On the other hand, you've got vascularization and blood vessels, which make the cancer grow and actually interacts and suppresses the immune system. So we built this meta signature, and a lot of work went into it. I'm making it sound trivial. And some of it was empirical biology and some was human curation. But we conceived of essentially a meta signature that captured these 2 biologies. We used an artificial neural network, a very simple, shallow artificial neural network, to model a real world patient data set. We only had 300 patients in that data set, but we had sufficiently few input genes that was okay. And we did a bunch of other stuff to avoid overfitting. But what we ended up with was this very simple conceptual framework where the model would classify patients into 1 of 4 subgroups. And if the patient ended up in the IA subgroup, we hypothesized they would do best against checkpoint inhibitors. That's immune active. If you ended up in the red A group in the bottom left, well, that meant you were highly angiogenic. You were likely to benefit from, say, an anti-VEGF like ramucirumab or Avastin. If you ended up in the bottom right in the ID group, that's the green group, well, your tumor microenvironment is pretty quiet from a gene expression standpoint. So maybe a vaccine is in order to kind of get things going. And the purple group, immune suppressed, is where things get really interesting because there's a huge industry of building drugs that are meant to serve as combinatorials with checkpoint inhibitors to activate. And so we built this model that predicts the subgroup, by extension predicts the outcome to certain classes of therapies, but also set up testable hypotheses, right? And so we could test whether this works, and sure enough, it does. I'm not going to share the actual response data of the model, although we've published it at a bunch of conferences and we're writing a manuscript right now. But I'll qualify the model in a different way. Both QIAGEN and Exact Sciences, 2 giant diagnostic companies, have each independently licensed the model from Ancserna to commercialize into actual companion diagnostics and research use only diagnostics, so point of care. And the Federal Drug Administration, the FDA, has approved this, not yet as a CDX because it has to do a prospective trial, but has approved it for investigational device use in clinical trials. And this is like seriously bleeding edge stuff at the FDA. This is a machine learning model of a gene signature with nearly 100 genes in it. It's a different kind of biomarker. It's one that embraces complexity and it works. At least 3 of those 4 boxes work really well. And I'm guessing the 4th one does too. All right, so the next problem is that the genes that you're putting in to train your model, the genes that constitute your signature, are going to have bias from somewhere. And all right, what do I mean by bias? So imagine that you want to build a signature. You've got some experimental evidence from, say, RNA sequencing data and other from microarray data. Well, if the gene showed up from one of those data sets and you put it in your signature, and the gene showed up from another data set and you put it in your signature, and so forth, there's a fair chance that gene behaves a certain way due to where it comes from, right? Due to the inherent bias from something to do with this data. It may not be the kind of analysis platform. It might be that some of the genes come from a model of colorectal cancer, whereas others come from patient samples of gastric cancer, right? Equally important to your biology, but harboring bias. And so you have to account for that. You have to look for it before modeling, and ideally select genes that don't harbor bias, but are robust across these. And so this is another really exciting area of research in our company. We've actually filed patents on this, where we set out to optimize the feature set before modeling to include only those genes that are robust to various potential sources of bias. And you could do this across as many sources of bias that you can think of, and also find data sets to evaluate. And so we were especially interested in the microarray versus RNA-seq, and for that matter, RNA-seq versus other kinds of RNA-seq, because we knew those were going to be the data types we were working with for the Young-Cerna model, and we knew that the actual clinical assay would require that. We were also interested in cancer type, so we looked at this across a bunch of different cancers, again, trying to find genes that would behave similarly, whether the disease was gastric or ovarian cancer or colorectal cancer or melanoma, et cetera. And we also looked at this in terms of the tissue source. So in the cancer world anyway, you could have fresh biopsy, but you might also have formalin-fixed paraformaldehyde embedded slides that could be a decade old. And we wanted to make sure that the genes were also robust to that sort of handling history. A really important first step. The last problem I'm going to mention is simply that models require external validation. And sometimes hypothesis testing, if that's the approach of your modeling. And in our hands, it is. So this seems like a gimme, external validation. It seems like this should be like, facepalm obvious, but it's not. At least it's not when you go look at those million biomarker papers in PubMed. If any of you actually do modeling yourselves as part of your research, you probably produced a figure like you see the one on the left. And I applaud you for your amazing area under the receiver operator curve score. 0.98 is excellent. What that says to me, assuming you did all the modeling right, you had a good, substantial holdout data set from your training set, and you did your cross-fold validation, is that your model is on the right track. It does not mean that your model is ready for people, because the next person you model that had nothing to do with your training data is going to blow this thing up. You've got to validate the model on independent data sets. And so in our hands, because we really like this idea of modeling biology and having a therapeutic hypothesis where we match some phenotypic classification to a drug, we need a bunch of data sets because we have to test all these hypotheses. And so then what you end up with is something like you see on the right, which is a whole bunch of batch effect. You get all these data sets that you want to test, but they're all over the place. And we can identify usually what the major effects are. So we know that, again, things like RNA-seq versus microarray or whatever is a big one. Sometimes, again, it's different kinds of RNA-seq or different kinds of tissue handling. We know that the origin of tissue has a big effect. And so, these are challenges that I think cause a lot of folks to stop with the figure on the left rather than actually doing the work that gets you to the point where you can have some confidence this model is going to work on people. And so what we've developed, the approach we've taken is, and again, we've filed some patents on this, systematic approaches, building systems that map those independent data sets into a coherent data space that allows you to do these independent validations, but not only the validations. Okay, great, you've shown your model works, but then you can take it to the next cohort of patients. You can take it into a clinical trial, or eventually, take it to the actual point of care, and it should work on the next patient and the one after that. And what you end up with, if you do this well, ideally is a stratifier, a patient classifier, that has a very clear decision boundary because it really is learning the signal, not the noise. So I love this hourglass figure as a way of illustrating, and these are real data points. It's nicely stylized. Responders in gold and non-responders in blue. It's not perfect. You get some of the responders at the bottom of your hourglass, some of the non-responders at the top, but you have a super clean decision boundary. You have a constriction where you can say with a lot of confidence that everyone up here are the ones that should get the drug and the ones down here shouldn't. And if you read journals like Clinical Cancer Research, where people publish classifier models a lot, what you very often see is a big area of ambiguity in the middle. Rather than an hourglass or a bimodal distribution, you end up with a near normal distribution of your model calls, where everyone in the middle is ambiguous and you can't assign them to one class or another. And I think a big part of it is the algorithm choice, but part of it is also losing a lot of the signal because enough care was not taken in the validation. Okay, that's my high horse. I'm going to get off of it. This is our product. I'm not selling software today, so I don't need to dive in too much further. But we've really thought a lot about this, and kind of this is what we do for a living. If you want to make it a little bit more kind of geeky, it's really a series of technologies meant for data flow. And so kind of building from the bottom up, building the signature through discovery data and ontologies and past knowledge, and doing all the good machine learning practice methods from model building. We're also super into visualizations and explainable AI, which I'd love to talk about if you guys are interested in that sort of thing. And then the validation piece, which I've waxed on about. Are there any questions, Melissa, that I should stop and address on that technical bit? Otherwise, we can get on to the stuff that's more interesting to people.
No, there are no questions in the chat box yet.
All right, don't be shy, guys. Cool. Okay, surfing the AI hype wave. When we were starting Genialis in 2015, and even worse, when I took over as CEO in 2018, we had already built our software that does all the data processing stuff. Now we wanted to go tackle real life science problems. And me being the resident life scientist, we felt I was probably best educated to do that. At the time, everybody seemed to be starting a AI for ... company in biotech. It was usually AI for drug discovery. And it's funny because there are a bunch of like newsletters and industry publications that track this sort of thing. And they just quit. It was like, I give up. There are like 200 new ones this month. I'm done. I'm not tracking this anymore. So there was absolutely an AI hype wave. It was annoying. A lot of companies would open their doors, put a .ai at the end of the URL and go raise money off it. And old timers, I'm not counting myself among the old timers. Remember, I was naive as could be, but old timers who had been around for a while were really wary because they had seen this before. They had seen it called other things in the past, but hype waves end up coming crashing down, and there's a whole lot of disillusionment at the other side where you can't get funded and you can't do your work, and you give the whole industry the promise of black eye. So around 2018, 2019, because we were serious about using AI where it was appropriate. And here I want to choose my words very carefully. We were serious about doing data science. We knew that machine learning would play a role in that, and we wanted to do it correctly. I hope I've convinced you in the last handful of slides that we're really interested in the boring problems. We're interested in the problems about things like data normalization and batch effect and crap like that. We wanted to do machine learning well. So we got together with a bunch of our friends in the industry, and we started an organization called the Alliance for Artificial Intelligence in Healthcare. January 2019 at the JP Morgan Healthcare Conference, we opened our doors with, say, 21 or 22 members, and that constituted the initial board. Today, we're well over 50 organizations. If you scan this, I think this is a bit of an old slide because I don't think there are 50 logos on it, but it's a pretty good NASCAR slide. You'll notice in the bottom left, Rice University. If you want to give her a huge headache, I encourage you to email my dear friend Angela Wilkins, who's the head of the Kennedy Center, who's Rice's representative. Feel free to just pester her all day long about this, but she should be able to tell you what's going on and potentially even find ways for you to get involved. You'll also see Google and AWS are here in terms of the big platforms. Some big pharma like Roche and Janssen. Some really, really innovative, smaller, newer biotechs. I mentioned Recursion earlier, in silico medicine, Cyclica. I mean, these are my friends. I mean, I love shouting out to them, but these are also the companies of the future, in my opinion. And if you follow the industry literature, you'll recognize some of these names. Some you won't yet. So we started the AAIH in some ways to play defense or to go on offense. But the point was to make sure we secured a future for the industry that we were trying to build that we were proud of. So we are the global advocacy organization dedicated to, you can read the tagline, discovery, development, delivery of better healthcare. But what we're really about is promoting for the responsible adoption of AI in healthcare. Now, granted, healthcare is a huge umbrella. AI is a big umbrella for that matter. There's a lot of different mathematical science bundled up there. We want people to do it right. And so a lot of the work we do, in fact, the only benefit of belonging, other than getting to sort of market off of it and have participate in some webinars and stuff, the benefit of belonging is you get to do work. You get to go to the meetings, you get to advise the FDA. Occasionally I'll take calls with Congress people who are on committees trying to do policy. We work with standards organizations trying to think about standards for both data sets and algorithms. We're trying to get it right. So this is a super cool organization. Again, I'm not trying to sell anything. I'm just sort of trying to illustrate my journey through this and also sort of get the creative juices flowing about the kinds of stuff you guys want to do, when you're not spending, Fridays at the Keck Center anymore, but out in the world. I think the Alliance is an awesome organization. I think it's awesome because our members are awesome, but we also have an important mission, and the work is really, really satisfying. The FDA did put out some more formal guidelines on its good machine learning practice for AI as part of software as a medical device, and you could see our fingerprints all over it. So that was really satisfying. Okay, so I think you should be able to tell that I don't think AI is all hype. In fact, I don't think it's even mostly hype anymore. I think it's pretty legit, and I think it's here to stay. I'm going to borrow a phrase from the World Economic Forum where they describe, and I don't think they're talking about AI specifically, but sort of digital technologies in healthcare as the fourth industrial revolution. And I like this. I tend to agree. So this section is exactly one slide of a list of a bunch of areas. These are domains that I am absolutely convinced are going to be disrupted by AI and related technologies. In some cases, we're already seeing it. In other cases, there may be higher activation barriers to clear. So the reason I'm putting this up here is I'd love for you to kind of scan the list and think about all the places you could consider working next or after that. And I'm sure I'm missing some, and I'm happy to dive into any of these to give examples of companies that are doing cool stuff or problems that are being solved or things that still need to be solved. I don't know all of them as well as others. I'll be honest, payers, like how you get reimbursed for stuff is still pretty opaque to me. But a lot of it I do know. And I just think that like all of this stuff, all of healthcare is inefficient. There's nothing about healthcare that's efficient. And the promise of your training is to be able to use data in a much more sensible way. Is to be able to connect people who sit on the domain expertise side and the data expertise side and make headway here. So your mountain is waiting. I don't know how many of you had someone read Oh, the Places You'll Go by Dr. Seuss at your college graduation. That's what this reference is from. But the idea is that with brains in your head and feet in your shoes, you can move mountains. And I think that the Keck training is especially powerful at this. I say this every time I get to talk to trainees, and I say it cheerfully, but also very grudgingly. I owe a huge debt of gratitude to Dr. Kabraki, to Lydia Kabraki, who, among other things, I mean, she kind of whipped me into shape in a lot of ways, but she insisted that I take a class on statistical machine learning. And again, I had given up on hard math when I was in early undergrad. I was a bench scientist. I was a scuba diver for graduate school, for crying out loud. Like I didn't want to do hard math. I didn't know how, but she insisted. And so I convinced, and I think Lydia had to intervene pretty seriously. I convinced Dr. Subramanian at Rice to let me audit her statistical machine learning class. Now, I couldn't do the homework. I wasn't good enough at math to do the homework, but I went diligently, took notes, and I understood most of what she was saying. And what that singular experience gave me, but also the kind of totality of my Keck training, is I became conversationally fluent enough to sit with one foot on both sides of the fence, to sit in the middle, to run an organization where half of our team are life scientists and the other half are data scientists, and to help these two groups of people communicate what are the problems and what are the solutions, and to come up with a shared language. And at the very least, that's what you get from the Keck training, aside from anything specific from your actual research and the network and the exposure. So what I'll leave you with is that your training will open any of those doors and probably others. And I reject on its face the idea that a PhD who has left academia has failed or quit. Now, that's not to say you shouldn't stay if that's what you love. Academic research is super important, but it is not the only thing. In fact, when you get out in the world and you go to Kendall Square in Cambridge, Massachusetts, you can't walk down the street without tripping over a PhD who is not in academia anymore. The academics are, well, I guess MIT is there, but academics are up in Harvard Square, everyone else is in biotech. If you go out to the Bay Area, same thing. And increasingly in Texas, you're finding that as well. So, think creatively about what you really want to do, but don't think too hard about it, because a happy accident was the best thing that ever happened to me. All right, I'm going to stop there. I'm happy to take any questions.
Thank you very much. There are not any questions in the chat box yet, but please feel free to unmute yourself.
So hi, my name is Violet. I have a quick question.
Sure.
You said you didn't know much about AI. You took the class to get a bit of an understanding and be able to communicate. How long was the road from there from like probably knowing some computational stuff like coding all the way to actually learning machine learning to the point where you are right now? Because that's a road that I would love to take, but it's very difficult to get there.
Yeah, I took a slightly different road. I took like the freeway feeder. I still don't know how to code. I don't have to code. Okay. That's what co-founders are for. With that said, let me quit being cute. I do recommend that everyone take some coding, even if that's not something you do. So while I was at Baylor, I did a class on statistics in R, and that was with who taught that? Sue Hilsenbeck, who's brilliant, by the way. Sue Hilsenbeck's one of the sharpest people in the world, and she's really good at what I didn't realize would become really important to me, which is clinical statistics. I didn't learn any actual clinical statistics in that class. I mean, I can read a paper and understand. I didn't learn how to do it, but I learned enough to understand, again, what people are up against. And then I took a class on Python and actually did write a program. It was 480 or 500 lines of code. And I gave it to Micha, who would become my co-founder, who's a Python whiz and prides himself on efficient software. And he checked it. And I guess there's a tool you can use called PEP8. You can run it against PEP8 community standards. Of the 500 lines, 480 violated the community standards for code structure and style. But it worked. And I even heard that some grad student years after I left Gaddy's lab had like patched it, updated the code so he could keep using it. It's just a tool to like help design cloning experiments. It actually was similar to something I'd worked on the bench side at Lawrence Berkeley. So anyway, the point is, my journey is just to learn enough to have these conversations, to understand what's possible. This is actually my favorite line whenever my son asks me. He used to ask me more often, like, Dad, what are you doing? Like if we're scribbling, if we're doing art, if we're cooking, I say, I'm just seeing what's possible, right? If you want to become a coder, you should pour yourself into it. If you want to just get better at coding, you should pour yourself into it. But you can do what I do without knowing how to code.
So actually, my work is computational biophysics, basically. So I code all day long.
Yeah.
Medicine was the one that I was lacking. However, I found that there is kind of a weird gap between
Mm-hmm.
the coding that I do, which is basically basic, so to speak, to get to the actual AI.
Mm.
Like I even took a class and I was like, I can follow the basics, but the moment you get into like the really difficult stuff, I am
Mm-hmm.
lost. And I've been coding for 6 years. And I was like, I have no idea what just happened. So that's why I was
Interesting.
curious about it.
Yeah, here's my guess. My guess is that I'm probably oversimplifying the vocabulary a bit, right? So machine learning and AI at the end of the day is mathematics, and a lot of coders are mathematicians, but not necessarily. Writing software and being a bioinformatician are very different things. This was something I learned. My company has learned the hard way over and over. We had great bioinformaticians. Most bioinformaticians I've ever met, and I do not mean offense to any of you in the room, are absolute garbage software developers. They're two different disciplines. They're just different disciplines, and that's okay. So, my advice, are you a grad student or postdoc?
So I'm a postdoc. I did my PhD in chemistry, but it was literally computational biophysics. Like that's one of the problems I'm encountering right now is I'm not a chemist, but I have a title in chemistry. I'm not a programmer, but I do programming. I am not a bioinformatics because I don't have genomics or anything like that, but I know about proteins, but I don't reach. I'm in kind of like a middle of a lot of different areas, and it's like, where do I go from here?
You join a team. So either you start a lab and you hire really smart specialists, or you join a company that's got specialists and you get to be the boss, or you get to be the linchpin for all of it. And I'm being serious. I mean, it's certainly in the realm of possibility that you'll choose to specialize and get really, really good at machine learning or something. But I don't think you have to. I think you just need to be part of a team. And it's a team effort. I mean, on our team, again, we're 30 people. We've got now four people who are really good at machine learning, right? But loads of people contribute to those projects and do different pieces of it. I hope I'm not underselling. We might have more than four. I hope no one on my team's watching and asks me who the four are. But anyway, the point is that we have bioinformaticians and we have software engineers and we have, data scientists and this and that. And I take for granted, and I'm surprised sometimes when we shift someone onto a new project, and I just assume because he's quantitative and he's a good coder that he's going to understand how to do stuff. But it's, these are all specialties, And I think being a generalist or being widely versed is great. I just think, don't beat yourself up if something seems hard. Like, just be part of a team.
Okay. Yeah, that's my main concern right now is I'm doing a postdoc here at UT Health, and it's like, this is great, but where do I gonna go from here? And like starting to look at some other options, and it's like, what do I sell myself as? Computational biophysics, chemist? So that's why I was wondering how you go there from that. But thank you for that, and thank you for the talk. It was really good.
You're welcome. I would say where you go next is again, you go to Boston and you work for top flight biotech doing the most amazing computational biophysics. I mean, right now, probably the most impactful area of AI in healthcare in terms of just turning shit upside down are people who are actually predicting new drug molecules, like building AIs that invent molecules that didn't exist before. And there are some rather mature examples of this already. In silico medicine has the first one of those in human right now that I'm aware of. You'll fit right in.
Thank you.
Other questions?
Oh, yeah, yes. My name is Kenny, and I'm also the postdoc fellow in the UT Health. And I have a question is because what we are doing the AI, and one of people, especially in the deep learning, and the people will be challenged to say, okay, this is like the black box, even though sometimes it's very powerful. And for example, is you can use the graphic neural network and combine the sequence and to predict some new molecules. And but the people will challenge to you, okay, how did you to convince people to say, this is maybe come true or something like the multiple evidence? So, I mean, in your company, when you do something like this very fantastic findings, how did you to sell to your potential customer to say, oh, this is maybe we need to invest money to do this because clinical trial or the drug development, like the meta compound, sometimes is very time consuming?
I think there are a few ways to answer this, and it depends on what the AI is trying to do. So a lot of the true deep learning that goes on in this space now is around that problem that Violet and I were just discussing, which is trying to design molecules or maybe target discovery, things like that. But that's pretty early preclinical stuff. And there, I'm a lot less troubled by having a black box, because undoubtedly you're going to have a whole bunch of assays. You'll have in vitro assays, cell line assays. There's a long way to go before it touches a person, and you can prove if it works. And you can usually prove if it works pretty cheaply and quickly. That said, we're all curious and we're scientists. We'd love to know how the model works. And so there are ever expanding toolkits for dissecting a model and trying to learn. But some of these deep learning networks, especially things like graph convolutional networks, graphs and graphs, they're just hard to interpret. They just are, and they may remain that way. The more hidden layers you have, the harder. When you go to the other side of the spectrum, where Genialis spends a lot of its time these days, which is more on the patient diagnostic side, then it matters a lot. You better believe the FDA is going to care what's going on inside that model, right? Patients are going to care, doctors are going to care. So you have a few choices, but you also have some constraints. You're likely not doing deep learning in the same context as you're thinking of it, in part because you just don't have enough patient data to do the deep learning on. Sometimes this is not always true, but it's often true, right? And in the case of the model I described to you, that was the training set was 300 patients, and it was a shallow artificial network. It had a single hidden layer with 2 nodes. It was the simplest artificial neural network you could conceive of by design, on purpose, right? Because we didn't want to overfit, and we knew we had to explain it. So in that case, and generally for these kinds of models, we have some very cool tools for doing what I call unboxing the black box, right? So for actually looking at understanding the impact of all the features, first of all, right? And so, there are methods like SHAP scores and things like that, for looking at the hidden layers themselves and projecting those into other spaces, so you can try to understand what the actual model is learning. And then for really interrogating the outputs in interesting ways. So kind of thinking about all layers of the neural network and systematically and comprehensively understanding what it's doing. Again, I think that's easier with simpler model architectures, and it's more important where simple model architectures are going to be the norm anyway by constraint. That's my experience anyway.
Yeah, I'm either.
Any other questions? I might throw one out in general, because you're working in biotech and it's not always stable, as you say. What kind of advice would you give someone about what kind of tools they need to work well? Because you might be coordinating a team internationally, you might be seeking funding.
That's a good question. I think things have gotten weird in the last few years everywhere, right? We're sitting here on Zoom, and you guys aren't going to get to have pizza in the courtyard after today's call and stuff. Let me start a different way. The most surprising thing to me about the world of biotech, and this is just because I had different notions. You probably know the names of many of the top 20 pharma, and they have big footprints in sort of obvious places like Boston and New Jersey and Basel and elsewhere. But there are something like 5,000 small cap biopharma in the world, and you probably don't know the names of most of them. Even once they've gone public, you probably don't know the names of most of them. And these guys start early, and they start like startups. They're kind of like tech startups initially, and they have small teams, and they try to raise a lot of money because it's expensive work. Literally just manufacturing drugs and doing clinical trials is very expensive work. But they can be done kind of virtually, just like a tech team, right? So it's not uncommon for us to meet a biopharma company that's maybe raised $100 million in venture capital, that's 10 people, and they sit in an office or in their home offices, and they outsource everything to CROs until such a time as they have a drug ready to go to the clinic, and then they IPO and they get bigger. That was interesting and surprising to me. Another way to answer this question is and this is kind of startup in general, but let's say biotech startup, because that's what we're talking about. My experience running a small biotech startup is what I can only imagine is what it's like to be a pre-tenure faculty, or at least what I've observed among my friends who are pre-tenure faculty. And let me tell you a little bit what I mean by that. You go from being a postdoc to a whole different job overnight, right? Like one day you're a postdoc, the next day you're not. And the day that you're not, you're no longer in charge of doing things at a lab bench or even writing papers. You're a bureaucrat. You're in charge of hiring people and finding money and hiring people. And so that's been my experience. It's not so different in a lot of ways to go from being a postdoc one day to being junior faculty versus being an entrepreneur. At least that's been my experience. I wonder if any of the junior faculty in the call would dispute that.
One last chance to ask Dr. Rosengarten any questions while you have him. All righty. Thank you so much for an amazing talk and for taking the time to.
It's been my pleasure.
And I thank everyone for being here, and I wish you a very happy weekend, and we'll see you here next week.
Parting thought, Melissa, feel free to hit me up on LinkedIn, anyone who's on the call. And if you want my email, Melissa can give it out. And I'm usually responsive, within reason.
It's linkedin.com//n/Rosengarten with a T. All right. Thanks so much. And thanks so much for
Thanks, everybody.
making yourself available. Bye, everybody.
Thank you.
Bye-bye.
Bye-bye. Thank you.